From Microbe TV, this is TWiN, This Week in Neuroscience, episode 41, recorded on June 13th, 2023. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hey, Vincent. Hey, everyone.
Is it really nice weather there?
It's actually been an unusual spring. It's been pretty warm, but also lots of thunder showers each day. So, yeah, I think this is the greenest I've ever seen in Utah. It's amazingly green.
Also joining us from New York, Tim Chung.
Hello. Hi, everyone. Great to be back.
And from New Orleans, Vivianne Morrison.
Hello.
Must be really hot there, right?
Oh, yeah.
Oh, yeah.
Yesterday, I really got to experience the heat because, I have time blindness. And so, the time to leave catches up, like it's surprised. I'm like, oh no, it's time to leave. I'm in the middle of something. So I had to sprint to my car to be on time to pick up my kids. So it was hot and I really felt it. Super sweaty.
I was there in April, I think, and it was already warm, but I just love warm weather. Being of Mediterranean descent, I just love hot weather. It's just hot and humid. Bring it on.
I lived in Phoenix for a few years. I'm not sure I agree with that sentiment.
Well, Hong Kong.
Hong Kong is hot too, but Phoenix is a different level.
So, Tim, I'm going to Hong Kong in November. What'll that be like?
Oh, November would be very pleasant, actually.
All right, you have to tell me what to do, okay? I'm going to take a few days and check it out, all right?
Okay, sure. I'll tell. It depends on what you like to eat. Mainly eating is good.
That's the only thing that's good? There's nothing else?
We have a Disneyland.
No. No.
Like a really small Disneyland.
Aren't there any historical things?
There's a lot. Yeah, I'll let you know. There is like the peak is very nice. There's a lot of historic stuff. Well, not too many, but like there's some.
Okay.
There's historical touristy attraction. Good for shopping, but actually food is the best.
All right, I like food. All right, we'll connect offline. But meanwhile, we have a paper to do here, which is very cool. People are going to like it. It's an Alzheimer's paper, and Vivianne's going to do us the job here.
Yeah, it's actually kind of very distantly also related to food because some of the neurons we're going to be talking about are involved in feeding behaviors. But this is a great paper. I think when I read the abstract, it's the first time in my career as a scientist where I stopped and I said, wow, that was a really well-written abstract. Like I immediately knew what was happening, the context and like what the takeaway message was.
Can I ask you, so, all right, obviously you have to be a neuroscientist to be reading this, right? Nature Neuroscience. But they say, first sentence, CA1 neurons. I don't know what a CA1 neuron is. Why don't they define it in the abstract? You guys all know what they are, right?
I actually think we should ask the neuroscientists who actually study neurons and like memory and learning and stuff. We should ask them to kind of break down the hippocampus real quick. It may have been done in a previous episode, but it has a couple of different regions. They're grossly associated with different types of information processing, I believe. But if someone who actually studies this could talk about it, I think it would be great to hear because then we could see like maybe why they chose to look at a particular region in the hippocampus.
I volunteer, Jason.
So the hippocampus, it has this very stereotypical structure, even in humans. And there's three main cell types and regions. There's the dentate gyrus. So I think the hippocampus is a C shape. There's two C's. One C is the dentate gyrus, and then the other C is the CA region. And so then you've got your dentate gyrus, you've got CA3, which the dentate gyrus cells project to CA3, and then CA3 projects to CA1, and that's this end of the hippocampus. And information flows into the dentate to the CA3 and then out of the CA1. So the CA1 is the output of the hippocampus. And it's famous for place cells and lots of different kinds of synaptic plasticity. So, that famous long-term potentiation was discovered in hippocampus. So it's a nice circuit because it's very well defined, and we know exactly what the input and output is of the circuit. And so the CA1 are the main projections out of the hippocampus, and so you often see memory-specific studies looking at CA1, partly because it's the output, and also because the kinds of plasticity and electrophysiological attributes are really well defined in CA1 because it's super easy to record in those cells, and it's very easy to get to know what the inputs are to those cells as well.
Yeah, and I think we're going to also talk. I'm going to call upon either you or Tim again to talk about if there's time and interest in talking about the EFIS methods that they use, those or like voltage clamp versus current, and then also maybe a little bit more about plasticity later on. But Tim, you were going to say something.
Just a very quick note. If you're wondering what CA in CA1 stands for, it stands for cornu aminis, which basically means it's like a Latin for some sort of ram's horn. So like a goat's horn.
And what does hippocampus stand for?
It stands for seahorse.
Right.
So there's a lot of animal
seahorse and horn, like ram's
but not
horn?
Jason's two C's, but C as in the Mediterranean Sea.
I mean, I guess you can't define everything in an abstract of a scientific paper. That's not the point. You have to assume that people in the field are reading it, like APP NLGF mice.
Exactly.
Right?
Yeah, and I will. I was going to bring this up later because I will talk about, just give a little blurb about this mouse line. But for the listeners who are interested, there are a lot of Alzheimer's disease mouse models, and they're all different, and they have different caveats and different strengths. So if you're interested in learning more about those, you can go to the Alzheimer's Forum. It's alzforum.org, and you can find information about all the lines. So yeah, the title of this paper is Early Alterations in the MCH System, which stands for Melanin-Concentrating Hormone System, link aberrant neuronal activity and sleep disturbances in a mouse model of Alzheimer's disease. So the first author is Dr. Sarah Calafate, and she is currently at a university in Portugal, but she started this work when she was in Belgium with Dr. Bart de Stroepe and Joris de Wit, who are the senior authors on this paper. And she's continuing this work, I think, based on one of the targets that they saw in some of the RNA sequencing that they did. So I'm sure there will be, and there's some questions that come out of, a lot of questions that come out of this paper, and I'll bring up a few of them that I think are interesting. But the take-home message of this paper, so that you can listen to this part and then go off and do something else, or you can stay with us and go deeper, is that early on in this Alzheimer's disease-like pathology that we can create using these Alzheimer's disease or AD mouse models, the neurons in the hippocampus start to go haywire. And the reasons for this are kind of TBD, but presumably it's because you have these neurotoxic accumulations of amyloid beta. And then additionally, there are changes in the function or the kind of condition of hypothalamus neurons that actually project to the hippocampus. And they're saying that the hypothalamic projections to the hippocampus is a mechanism for trying to settle those hippocampal neurons down to some set point of activity. And little side note, the narrative is told such that as the reader, you kind of unknowingly start to operate in this context in which the hippocampal neuron dysfunction happens first, and that the hypothalamic neurons are then kind of recruited and responding to this dysfunction in an attempt to compensate. but I'm not 100% convinced that they conclusively show that. But intuitively, it makes sense. So anyway, they find that this compensatory reaction works for a little bit, but then it fails. And they identify that it fails to like suppress neuronal hyperexcitability in the hippocampus, and which correlates with the kind of cognitive decline, learning and memory deficits that we see with these AD mouse models. And they identify a gene that gets turned on around the time that they see this compensation starting to happen. And the gene codes for MCH, the melanin-concentrating hormone. And so then they try to dig into how would MCH actually influence neuronal activity. And then they ask whether or not this is the or one of the mechanisms by which the brain tries to counteract the hippocampal neurons starting to become dysfunctional. And then also the narrative suggests that this failure of the compensatory mechanism is a result of the fact that the neurons in the hippocampus, their hyperexcitability is either increasing, worsening, or just continuing. But they also show at the very end of the paper that the MCH neurons themselves, those that they are dysfunctional. And in the way that I interpreted it, the hypothalamic neuron dysfunction is maybe cell autonomous, meaning that it's not a functional or synaptic response to the dysfunction or the like inputs coming back from the hippocampus, because I believe there are hippocampal hypothalamic projections that go the other way. And we often see these feedback loops in the brain, but I'm not sure that that is at play here. But I think an unanswered question is whether the hypothalamic neurons are subject to the same instability that affects the hippocampal neurons, or if there's some other mechanism of dysfunction. All right, so we're going to talk a little bit about background now. So in Alzheimer's disease, I imagine actually a lot of people have heard about this, but there's something called a prodromal phase, which is just that like there's something occurring that you might not be able to see or see in its full-blown manifestation that's occurring before the actual onset of the disease, before the clinical diagnosis can be delivered. And so we've been able to explore some of this using these mouse models.
I would say in the humans, I mean, this is what makes it quite hard to treat Alzheimer's disease, is because people come into the clinic when they actually have symptoms already. Mm-hmm. But by that point, the disease has been ongoing for a decade probably, in that these toxic proteins, the pathology is accumulating. And so these drug trials that have been ongoing and, as you know, many have failed, partly because they are intervening in the disease too late. Mm-hmm. And so if we could figure out how to diagnose people early, or even sort of figure out, what drugs to use in the early phase, that I think would be really critical, would be really impactful.
So there's no, for example, the deposition of these amyloids, you can't sense that in the blood at all. That's completely restricted to the CNS, right? There's no
people are looking for biomarkers, but I don't think been.
You can actually detect these, like beta amyloid. You can detect in the blood, and there has been some clinical trials looking at tau and is the other toxic protein that you can measure, and there's some other sort of neuronal proteins that you can detect in blood. So there are some studies showing that you can use these as a biomarker, and I think that's probably where the field has to go in terms of sort of being able to really treat this.
Yeah, that crossed my mind. I thought about is, MCH, could it be used as a biomarker somehow? Because the hypothalamus is one of the places where there's relative permeability of the blood-brain barrier, because it's connected to, it either generates or is connected to the tissue that generates a lot of the hormones that are then secreted into the blood and or released into the blood. So I was like, maybe MCH could be used as a biomarker, but it's just not always so straightforward. You can have levels, you can have, for example, like folate. You can have totally normal external, like peripheral blood levels of folate, but have a folate deficiency in your brain. Like how that works is anybody's guess. And so, I definitely think somebody should look into this. Like, to what extent do changes in MCH neurons that are projecting to the hippocampus, how are they reflected in peripheral MCH levels or in some other system? But it's just really hard to tell what that's going to yield.
Yeah. And the other thing is a big point that you sort of mentioned here and that they concentrate on is that there's this equilibrium in the brain. There's always homeostasis. And this is really important for synaptic transmission, where there's many, many different processes that keep the cells in your brain in an optimal window of activity. And of course, if you have dysfunction in that, you get things like epilepsy, where your circuits are completely haywire. But it looks like in Alzheimer's disease, this prodromal phase happens where there's this long period of balancing the toxicity that's happening. And so there's a lot of plasticity in the brain, and it can compensate for this until it doesn't. And that's where they're coming in and saying, oh, what are the homeostatic plasticity mechanisms that help prevent neuronal circuits going crazy? And they're good for a certain period of time, but then they're not good.
Yeah. And actually I'd be, I wanted to talk about what that compensation looks like at a level that we people, everybody can kind of appreciate. So there's a ton of fMRI studies or functional MRI studies that look at brain activation patterns when you ask people to do tasks. They might be focused on verbal processing or auditory information or visual information processing. And they have found, or many of these studies have found, but the interpretations and the conclusions are somewhat debated. That, for example, during a verbal processing task in cognitively intact adults, the high performers. So typically, this task would recruit largely your left hemisphere, but the top performers in those tasks were actually individuals whose fMRI scans showed recruitment of both the left and the right hemisphere, which might seem like a good thing, but it's arguably not, because it means that it could mean, one of the interpretations is that the function of, for example, the left hemisphere, and I'm speaking extremely broadly, but that the function of the left hemisphere is somehow reduced. And so then the brain has to, in order to succeed at completing the task, recruit other brain regions. And this has been found, some similar things have been observed in people who have genetic risk factors for Alzheimer's disease. So probably a lot of people have heard about the ApoE4 allele, and that if you're, I don't remember what the percentages are, but even if you carry one copy of that, you have an increased risk of Alzheimer's disease. And so I just want to read a little blurb here from a nice review it was published in 2008. So I'm sure there are other papers now, but it's a review by Han, Bangan, and Bondi. And they say that in a challenging auditory-based memory task, individuals carrying that ApoE4 allele demonstrated a greater magnitude and extent of activation in regions that were outside of what would typically be used for that task. And in particular, it was areas that demonstrate increased activation as cognitive efforts or cognitive load increases, even in normal adults. And then what was interesting about those studies that they were citing was that there were longitudinal findings. So they would check in 2 years later with the individuals who were tested. and it was actually associated with memory decline. So there's definitely a relationship between like trying to squeeze as much as you can out of your brain until you can't anymore. So, and I think similar things have been noticed in individuals who speak 2 languages. Their prefrontal cortex is kind of like, that muscle is strong. It allows them to switch between the 2 languages. And while those individuals might be able to squeeze more, oomph out of their brain and combat cognitive decline, as soon as that, whatever that compensatory mechanism is, as soon as that starts to go down, there's rapid onset of cognitive decline. And that's why it can be so like shocking, especially, like what Jason was saying, there's, once it's obvious, there's not really much that you can do to treat it. And I think that's even more so the case for individuals like this who may have like strong compensatory mechanisms, but that just leads to like probably a more rapid exhaustion of whatever resources the brain has. So this is maybe like a prodromal sign, right? If you see these fMRI scans lighting up more brain areas than you would expect for a given task, but like we can't all be walking around, going in and getting our fMRI done, every 2 years or whatever. But there are other things that are associated, like signs that are associated retrospectively with Alzheimer's disease, like loss of smell, gastrointestinal symptoms. Like I think constipation is often associated with neurodegenerative.
Hey, those are Parkinson's prodrome.
Yes, they are. They are. I mean, it's a neurodegenerative disease. There's a lot of similarities. So, and then also sleep disruptions, which they bring up in their introduction. So, so this disrupted sleep, it's actually it's neuronal hyperexcitability. And it's not maybe quite the same thing as that increased brain recruitment, but it's probably related. So, as you can imagine, if a neuron is like hyperexcitable, I imagine it might be energetically expensive for the neuron and perhaps beyond kind of whatever like buffering capacity, so to speak, the neuron has, even when because it has a finite amount of resources, even when it's, has astrocytes and oligodendrocytes supporting it. So it's probably like tough on the cell, but it also, and then it'll disrupt brain oscillations or patterns of activity that we know that show up in sleep, for example, that we know are important for learning and memory consolidation. And then later on, cognition during wakeful periods.
But do people think that in Alzheimer's disease, neurons die because of too much excitation, like excitotoxicity due to increased calcium, that kind of stuff?
I'll have Jason talk about that, and I'm sure the answer is yes. But I think there's a growing recognition that those neurons are not yet dead. So like we see reduced activation in the hippocampus or in the temporal lobe, and that's, people are like, oh, the cells are dying. And of course, there is like, we know that the brain becomes atrophied. But that I think is just as much a result of loss of the neuropil of like synapses and dendrites. And I don't know like how much, like, is it equally that and cell death? I don't know, but those neurons may not be responding, or they may not be able to like leverage the mechanism that we use for fMRI is based on glucose uptake. And there's increasing evidence that individuals who go on to develop Alzheimer's disease, their neurons are not able to process glucose as well. I don't know like how or why, but so I don't know that they're dead necessarily.
Yeah, I think so. It's certainly the case that it seems that synapse loss correlates better with the cognitive dysfunction that you get early in the disease. But of course, by the end of the disease, if someone is still alive, you get worse and worse. It's not just dementia; it becomes essentially you become incapacitated, and that's because you have cells that are just dead. But whether it's because the eventual reason why the cells die, it seems pretty complicated. I mean, I'm not sure. There was a large push for excitotoxicity as being the all for neurodegenerative diseases, but none of those drugs ever worked.
Right.
So I would say it's probably complicated, but it's definitely true that losing the synapses seems to be the sort of initiating dysfunction, and then everything sort of cascades from there.
So I wanted to talk a little bit about sleep, because that's, the neurons that they talk about that come from the hypothalamus are recognized as big influencers in sleep, and how that relates to memory, and then how it relates to Alzheimer's disease. So sleep has for a long time been recognized as an important part of memory consolidation. And memory consolidation, at least as I understand, is essentially the process of creating these more or less stable cortical representations of information that was recently acquired via the hippocampus. And there was another recent paper that created a model to understand how the hippocampus and the cortex talk to each other, and try to understand, what is happening in sleep, what signals are going between those two brain regions that then leads to like effective memory. And so I'm going to read a quote because it just says it better than I can say it. Oscillations, so brain waves, are leveraged to support learning that leads to useful changes in memory representation and behavior. Their model has a non-rapid eye movement, or NREM, sleep stage, where dynamics between the hippocampus and the cortex are tightly coupled, with the hippocampus helping the cortex to reinstate high-fidelity versions of new attractors. I'm not sure what attractors means. I didn't look that up.
I think there's two things about sleep. One is that you need it for memory consolidation. How exactly that works, I think there's a lot of work still to figure out. The other sort of side of it that I think has been really interesting and more of a recent development over the last 5 years or 10 years is the idea that during sleep, you reset the brain both at a metabolic sense as well as in terms of your synapses, in that there's again homeostatic plasticity that's happening that have sort of resets the circuit so that they can learn again. As part of that, though, there's also this idea that you clear out all the junk that accumulated during the day when you had sort of this high activity hypermetabolism, and so that includes A beta and these toxic peptides that may get cleared out during sleep as well.
Mm-hmm. Yeah, it's something that you just said reminded me that these MCH positive neurons in the hypothalamus are become activated when there are high levels of glucose in the blood. So I'm like, oh, is that why? It's not just that all of the blood gets diverted to your gastrointestinal tract after you eat a big meal. Maybe it's that your MCH neurons, which are involved in sleep induction and maintaining sleep, they're just getting hammered by all this glucose. Okay, so yeah, I think it's super interesting that there's this kind of talking back and forth between the hippocampus and the cortex to help that happen, and that maybe you have the hypothalamus or other brain regions that you're like, oh, I never saw that coming, that are coming in to try to help mediate that communication.
Right.
So just a quick word about the hypothalamus. It basically controls everything. So, whatever you're studying, you should probably look at the hypothalamus. Oh, and also, excuse me if I mistakenly say hippocampus when I mean to say hypothalamus, or the reverse. Please, point that out, because it's just really easy to mix them up. But yeah, so the MCH neurons, they're involved in eating patterns and metabolism and periods of wakefulness versus sleep, mothering behaviors, lactation, the stress response. So yeah, it has a big role and these neurons specifically play a large role in coordinating the body and the brain so that, you can be effective and efficient.
But in case the listeners are as confused as I am, because these neurons make this thing called melanin-concentrating hormone, and immediately you think, oh, that's my skin color when I go for a tan. Apparently it only changes skin color in fish. That's where it was first discovered. And then in mammals, it seems to be mainly in the brain. And we don't know its function in skin yet.
Apparently. Well, isn't there there's some crossover between the metabolism of melanin and either, I'm trying to think if it's like neurotransmitters or... Anyway, this is above my pay grade. I don't do this.
Melanin is not related to melatonin, right? A lot of people are going to make that connection.
Yeah, I don't think so, but...
I don't think so, yeah. I'm not sure.
Okay.
But there is some... There's this thing called neuromelanin, which is a melanin-like substance. So I'm probably wrong about this, but there's this thing called neuromelanin, which is a melanin-like substance in the brain that is essentially oxidized dopamine and norepinephrine. So those are probably related structurally.
Yeah.
But in the skin, it is melanin, and I don't know how... Yeah, I'm not sure how that relates to the brain, except for melanin-concentrating hormone seems to be taken up in mammals and put in the brain. Bottom
line is, for not talking about melatonin, although if you have trouble sleeping, maybe you should take some, but that's another episode right there. So before we get into the data, I just wanted to point out why they use the mouse that they use. So Vincent mentioned this is the APP-NLGF mouse model of Alzheimer's disease. And in this particular model, they're not overexpressing or inserting multiple copies of a human gene. Like, we humanize these mice because mice don't typically, they don't develop Alzheimer's disease on their own. We have to actually introduce the human version of the genes that are involved, and usually they have these mutations in them that have been associated or identified in humans and are associated with familial Alzheimer's disease. So in this particular animal, we don't have overexpression in the sense that we're not introducing multiple copies, but the APP gene has a couple of mutations. There's 3 mutations that lead to increased accumulation and increased synthesis and production. So it's kind of, I think it's that much closer to like something that's more physiological or more relevant or real, rather than just like we shoved a bunch of copies of this one gene into a mouse brain.
Yeah, no, that's been a criticism of the Alzheimer's mouse models, that you're basically driving so much expression of a human gene, and so this knock-in approach where you just knock in the mutations into the endogenous gene is a lot more physiological. But the key is that this mouse model still develops similar amyloid plaques and pathology to those transgenes. There's debate over what model recapitulates Alzheimer's in a mouse, but in the end, they're all just models.
Yeah. Something else this was on the alzforum.org, but I saw that they say neurodegeneration is not detected.
Right. And so most of these mouse models... if I say
neurodegeneration, that's me misusing the word, because the authors did not use that word in their paper.
Yeah, but that's key. So almost all these mouse models, you actually don't get cell death. Right? So that's the neurodegeneration says the cells die, and that's the end point of the disease. But most of these models, there are very few of them actually have cell death. And partly I think it's thought because their lifespans aren't long enough that this kind of toxicity in humans, like we just mentioned, takes a decade for it to start manifesting, whereas in mice it's just not, they die after 2 years. But yeah, it is true that cells are not dying.
Yeah. And just to speak to that, like, reduced timeline, we start to see that prodromal phase in the Alzheimer's disease mouse models is like 2 months old is when you start to see the protein aggregates. And then, then you won't see cognitive decline, though I think you'll see some behavioral changes. But then, at like 6 months, then you start really seeing kind of pervasive disease at the tissue and cellular level accompanying the cognitive deficits. So the data. Okay, so they needed to first characterize or to show that the mouse model that they were using did have neuronal excitability, hyperexcitability in the hippocampus, which we've seen in other models, and it's associated, there's indications of that in humans. So they just did some ePhys, electrophysiology at a couple different time points. And like I said, it's that reduced time frame. So the animals were 1, 2, 3, 4, and 6 months old. And they did find an increase in the frequency of spontaneous activity at 2, 3, and 6 months, but not at 4 months. So they guessed that maybe there was this compensatory mechanism coming online at 4 months. And then also at that same time point, they saw that there was a small but significant decrease in the amplitude of those postsynaptic currents. So the neuronal, output of those neurons, again, pointing to this plasticity, and it's the homeostatic plasticity that Jason mentioned earlier, which is like trying to keep the neurons within some or around some set point in some window of like optimal firing. What that is, I don't know, and I don't know how you determine that either.
So the key to this technique, this patch clamp technique that they're using, is that it's super sensitive, and what they're actually measuring here is called a mini EPSC, which basically is the minimal unit that you could really quantify at the synapse. So these are all synaptic recordings, in that one little blip, and you can see in the recording, is called a mini, and that's thought to be quantal in that it's like one synaptic vesicle released, and you can that it's activating the receptors at the postsynaptic side.
And this is in contrast to like evoked potentials that are like we're releasing a ton of vesicles all at once.
Right, exactly. So this is a single. It's basically able to. It's sensitive enough to record from a single synapse, even though you're just you're recording from the whole cell, but the blip is a single synapse, and so the amplitude is thought to measure that postsynaptic side, so how many receptors are in there, and then the frequency is thought to really measure how many synapses there are there, and or some other things that you can interpret. But anyway, but that's the nice thing about this, is that it's really sensitive. It can detect changes at single synapses, and is the sort of best way we can, record synaptic transmission.
Yeah, and so they complement that with something that doesn't have quite as much resolution, and that's just they did some immunostaining for an immediate early gene that probably has come up on this podcast before, called CFOS. And yeah, so they found that there was a transient decrease in the number of active or CFOS positive neurons in the APP mice relative to their wild type controls at that 4-month time point. But there was it was a little unclear in their histograms. Like, I mean, we don't have to get like too much into the weeds, but like they didn't see an increase in CFOS positive cells at 6 months, which is when they're saying the compensatory mechanism fails to kind of keep neuronal excitability lower, closer to that set point. So they didn't detect an increased percentage of active neurons, but they did see an increase in CFOS intensity per cell, which I'm always like immunostaining. Like, I know it's well accepted. but like quantification of immunostaining can be, if you're not like counting cells, if you're actually measuring intensity, just can be really tough. So,
yeah, I agree. I think the electrophysiology in this first figure is way more not believable, but it's more impactful. I think CFOS staining tells you maybe a little bit. So it can get complicated in that you have synaptic transmission homeostasis, but you also have something called intrinsic excitability homeostasis. So that's the set point in the cell that whether it's basically easier or harder to have an action potential, and that intrinsic excitability could influence CFOS expression inside the cell. So you could have more CFOS inside the cell, but that may be a compensation for the synaptic transmission that's happening, the deficits, and so that's where you could have this teasing out of more cells getting CFOS activated versus more CFOS in the cell.
Interesting.
Yeah, and in fact, at 4 months, these cells that are compensating, they actually have very intrinsically unexcitable. This is in the supplemental data. So you can't really get them to fire very well.
Yeah, and they also looked at the number of spines and found that the density of spines was reduced. So that's going back to what Jason was saying earlier. So they're like, okay, we see that this 4 month time point is interesting because there's something happening there to reduce excitability. So they use this cool technique called spatial transcriptomics and they identified the gene that codes for the MCH protein. And they find that it's upregulated at 4 months, which coincides with that that transient normalization. And if I understand spatial transcriptomics, it's like you overlay your tissue on essentially like a little grid, and each one of those squares has the squares of the grid has a barcode, and then within each square there's a bunch of primers that would recognize whatever transcripts or mRNA molecules were present in that region of the tissue. And so then if you see that one particular square, like boom, there's a peak of amplification there, meaning that there were a lot of that gene was being produced there or being turned on, then you can use the barcode and be like, oh, okay, yeah, that's in the this layer, this region. But they looked specifically at the CA1 region, at the pyramidal cells. And they were like, oh, this is interesting. We see an increase in the transcripts for the MCH molecule, but the protein is not expressed in those neurons. So it turns out that what they were detecting were transcripts that were in the axons of neurons whose cell bodies were in the hypothalamus. And so those were the hypothalamic neurons were projecting to the CA1 region. So that's what they were detecting, which I think also speaks to the sensitivity of this method.
Yeah,
because that's crazy.
That's really cool. Because I think there's been a lot of arguments about whether protein synthesis takes place in the axon. Because there's no like rough endoplastic reticulum where all the ribosomes are normally studded on in the axons. But people keep detecting interesting mRNA transcript in the axons that suggests there is translation going on in the axon. So I think there's increased evidence that there's translation going on the axons.
Right.
Yeah. No, I think that's in itself an interesting finding. And I mean, this is a neuropeptide as well. And so there's really not a lot known about how these neuropeptide neurotransmitters or neuropeptide hormones are made and released from cells. So.
Yeah, and I think also I read that, let's see. So the MCH protein also contains 2 different neuropeptides on their own. So if potentially there's some sort of cleavage process of MCH happening in that presynaptic neuron, the hypothalamic neuron, which that cleavage process could also be regulated in a context specific way, then whatever that MCH positive neuron is putting out could be different. And I did see a recent paper saying, bringing up the possibility that these MCH neurons don't actually have like a neurotransmitter identity that we can like for sure know what it's producing, that what they produce may actually be context dependent. So I think in one of the papers, they actually were able to detect GABA and glutamate being released differentially. So that just adds another layer of complexity to the situation, and I think speaks to the ability of the brain and individual, like little locations and circuits, to fine tune things to achieve some goal or to offset some external event or neurodegenerative process, which is also just like super fascinating. But anyway, so they're like, okay, MCH is being upregulated by these hypothalamic neurons, projecting to the hippocampus at a time when we see this AD-related hyperexcitability being normalized, suppressed, calming things down. So how would MCH accomplish this? So they do a series of in vitro experiments as well as a slice culture experiment. So they just to see what does MCH do to the firing patterns of wild type hippocampal neurons. So they culture the hippocampal neurons, they treat them with MCH, and they see that the MCH treatment reduces the frequency and amplitude of those minis that Jason mentioned earlier. And then in addition to looking at just, doing those patch clamp assays where they look at individual neurons, they actually culture the neurons on a multi-electrode array where you can get like a bunch of different, we can do like field recordings where we're looking at a whole bunch of cells all at once, but we lose the resolution of like what's happening at each cell. So with the electrode array, you get a little bit more resolution. You can see what's happening at like individual places on the array.
Basically, it's recording action potentials from many, many cells. So sort of the absolute output of the cells.
Okay.
Just out of interest, so we haven't quite talked. I can't remember. We did talk about a little bit about MCH involving in sleep, but do you think these slices of hippocampus cultured on an electrode array, does it sleep?
No,
not at all. Because sleep is, defined by oscillations. And you can't really have oscillations without.
Yeah, you've cut all the inputs and outputs to the circuit, the rest of the brain.
Right. So with that caveat, it does look at what happens if you dump a bunch of MCH on the brain.
I think there is a way to prepare the slice cultures where you can keep intact some of the connections but they're like, within the hippocampus. So
these aren't even slices that they're recording. This is cultured neurons.
But they next do a slice culture prep. But yeah, what we're talking about right now is just cultured neurons. So they're just, they take a piece of, they dissect out the hippocampus, they chop it up physically, digest it, enzymatically, and then they spit all the neurons out onto a plate. And then the neurons grow and start forming connections. And then you can measure their activity and how they respond to different treatments. And getting back to something that Jason mentioned earlier about like looking at specific spines and like what's happening postsynaptically and, what we can say, what these measures can tell us about receptors. They see this, reduction in activity, and they do an assay where they can look at the amount of phosphorylated glutamate receptor versus total levels of the glutamate receptor doesn't change between the treated MCH treated and non-treated. But what does happen is the amount of phosphorylated receptor goes down, and that is the phosphorylated version is involved in retention at the membrane. So what the interpretation then is that MCH treatment is it's binding to something that is reducing the phosphorylation or leading to dephosphorylation of these glutamate receptors at the postsynaptic face. And that's leading to their internalization, making that postsynaptic neuron less sensitive So reducing its, firing rate and frequency, amplitude, blah, 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 neuron stuff. And so then also interestingly, like they find that that MCH is like even more of can be a modulator. Like it can reduce activity. Like if you treat the cells with the MCH, it reduces their mean firing rate, the neurons. But then they also show that like if you drive neuronal activation even lower by suppressing them with this GABA agonist baclofen, that the MCH treatment actually keeps the neurons at that very low level. So I don't really know how to put this into words, but that's interesting. That it's like if you have some extrinsic event or agent that's bringing down neuronal activation, then the MCH can actually keep it there, which I don't know, that feels meaningful to me.
Yeah, so one way to think about this homeostasis, right, is that what the again, it's that window of synaptic transmission and that you want to keep in the same ballpark. And so a homeostatic, so let's say you increase activity, what you want to do for a homeostat is ultimately decrease that to a set point.
Mm-hmm.
And that's what's happening in this Alzheimer's prodromal. You get high activity, something needs to bring it back down. And that's what they're hypothesizing, that this MCH is bringing it back down. And the opposite can happen, where if you decrease activity, they want to
go back
the set
up.
point wants to go back up. And so that's where it's interesting here, where
it's both homeostatic and disrupting other homeostatic mechanisms, right? It can be homeostatic in and of itself, but then it can also counter other homeostatic mechanisms, right?
Yeah, and that's just because this is a sledgehammer. I mean, you're just dumping on a whole bunch of MCH.
True.
So I wouldn't, that part of it's a little more not really physiological. But I think that it's going in the right direction that you would expect for it to counteract hyperexcitability.
Mm-hmm. And I think your point about like it's a sledgehammer is a good one. And the use of the slice cultures is like a kind of, it's one step closer to like a full brain. And obviously it has its caveats.
Those are acute slices that they're recording.
Right. So maybe culture is not the right the right word. But I do slice culture. So I think I'm just used to saying that word, those two words together. So the acute slices, what was interesting to me also is that like when you take the wild type brain slices and you put MCH on it, you don't see any change in neuron excitability. So I think that but you do, importantly, critically, you do see an effect of MCH on the APP acute slices. So you put on the MCH and you see that the hyperexcitability is reduced. But I just think that that's interesting. The wild types, you don't see a difference in the slices, but you do in the cultures, because it just speaks to like even within that slice, there's a lot going on that is trying to keep everything within some optimal window.
So presumably the APP protein in those mice is doing something to the MCH effect, right? Either directly or indirectly, because that's the difference between the wild type mice and these APPs, right?
Right. So,
yeah, it's complicated because at 3 months, that is when the homeostatic clamping down of MCH kicks in.
Mm-hmm.
And what you see like in those brains is that the MCH protein is upregulated, but now they are putting in extra MCH and show that kind of postsynaptically there's a kind of exaggerated response as well. So what causes the exaggerated response kind of postsynaptically? I think it's not too well known.
Yeah, I don't.
Yeah, but I think as Vivianne said, there's just a lot of processes that are happening. So MCH is just one contribution to the homeostat.
Yeah, and I think something. Go ahead.
All right, but something that might address Vincent's question is that they also looked at what happens if you just, so imagine, remember these APP Alzheimer's mice that have had increased exposure to MCH because of this compensatory homeostatic regulation. And they showed in the kind of a panel above that if you just dump MCH in a normal mouse, you can affect a lot of gene changes. So maybe there are all these gene changes going on in the background already that allows them to respond more to further MCH.
Were those animals treated, or were those hippocampal neuron cultures?
I think we're overcomplicating it here. I think we.
Anyway.
Yeah, that's because I think we're going to move on a little bit. But I think the key is that you have this homeostat, and MCH seems to act in a way that compensates for the hyperexcitability. But then at some point, it stops doing that, and then that's the next question that they're trying to address, right?
Yeah, they want to look at
what
those.
Actually happens to those cells that make the MCH.
Yeah, and I would say we have that was it's superficial still, because what Vincent's question made me think about what I'd said earlier, which is that we don't actually know what's happening to the MCH neurons. I would guess that I'm obviously not a gambler, that there's something that's happening to those hypothalamic neurons that maybe, is not mechanistically similar to what's happening to the hippocampal neurons.
Yeah, right.
And whether that is because, and I don't know that it's because of amyloid deposition. Might be, maybe not, but anyway, yeah. So they wanted to look a little bit more at the MCH neurons, and this is where sleep comes into the picture. And these are actually really cool experiments. So we know that the MCH neurons influence sleep, and then and they're maximally activated during REM, or rapid eye movement periods. And in that same region, kind of like sprinkled in, are these orexin expressing neurons, which promote wakefulness. So they wanted to look at kind of like the balance of activation of these two cell populations at different stages of sleep, or not different stages, but different sleep paradigms. So they have two paradigms here. One is sleep deprivation only. So the animal has 6 hours of sleep deprivation, and then the endpoint sample is collected. And then they have a rebound condition, which is where the animal undergoes 6 hours of sleep deprivation followed by 4 hours of rebound sleep. Then the sample is collected. And so in wild type mice, it's been shown that in the rebound period, the MCH neurons become highly activated. But interestingly, they find that does not happen in the Alzheimer's disease mouse. So the orexinergic neurons, they will go down during this rebound period. And that does happen in the Alzheimer's disease model, but it's really those MCH neurons. The increased activation during rebound sleep that we would expect to see does not occur.
And just to clarify, these are Alzheimer's mice at 6 months, when it's been shown that the compensation mechanism is falling apart. Is not able to compensate anymore.
Mm-hmm. Right. Which again brings it back to but why? What's wrong with these MCH neurons that's preventing them from becoming activated?
I would say I would have liked to have seen a similar set of experiments for earlier time points, but
that's a good point. Because what if you have even, it's okay, this is the other thing I was going to say. I looking at the pictures of the orexin positive and MCH positive neurons. The pictures we often talk about trying to pair your pictures with your histogram so that they look, they're talking about the same thing. If you see a 50% increase in something, you want your pictures to show a 50% increase. That's what it looks like. But the pictures in this it was a little bit confusing. And in some cases, I wondered if maybe the Alzheimer's disease mice had fewer MCH neurons and orexinergic neurons, but it wasn't consistent. So it just leaves me wondering can I just get a neuron count? Are we seeing cell death in this region? Or is it the same number of neurons that are there, but is their identity changing? Like they're no longer producing MCH, so you wouldn't pick them up. Like what percentage of the CFOS neurons were not, MCH or orexin positive? So anyway, getting into the weeds, but that's what I think about at night. Basically, they're showing that this kind of adaptive MCH increase that we see in wild types is not happening in Alzheimer's disease. And because MCH is involved in sleep induction and especially REM, they wanted to then look at this, what they call the sleep architecture. So that is how much time in their paradigms, how much time was spent awake in the non-rapid eye movement period and then the REM period. In with respect to the that 4-hour rebound period that they're allowed to have. And so they find that the wild type and the AD mice spent comparable amounts of time of their rebound period awake, which is about 20%, comparable amounts of time in non-REM, which is it's about 60%. And that non-REM is including like all those different power bands that we talk about. You hear gamma, delta, alpha waves, slow wave sleep, light sleep, all of that. And then the number of times that the mice entered REM sleep, because we know MCH is involved more heavily in REM, that was also comparable between groups. But the Alzheimer's disease mice spent a smaller percentage of that rebound sleep time in REM. And it's a couple percentage points, but it's significant. But something doesn't really line up for me here because there's like a piece of time that's not accounted for somehow. Like if they spend, within 4 hours, they're awake the same percentage of the time, they're in non-REM the same period of time. The number of times that they entered REM was the same, but then the REM time is a little bit less. I'm like, where's the rest of the sleep time? Did they sleep less?
Yeah.
Do you know what I mean? And I'm not like sure. I was like, maybe I'm just bad at math.
They're not measuring. So percentage of time wake.
Mm-hmm.
I don't know if in supplements that they looked at like slow wave sleep or non-REM sleep.
Yeah. They did a little bit.
What they're counting as non-REM sleep, I guess.
Yeah. So I wrote myself the same note, which is like, maybe future paper is going to be breaking down the non-REM. And the reason that I say that, they didn't tell us like the total amount of, they didn't like show us a graph that said like the total amount of sleep in that 4-hour period. But like, they kind of indirectly said that the animals spent the same amount of time asleep because the two groups spent comparable amounts of time awake. So, I'm like, I don't really understand. But
maybe these mice have a type of sleep that is a third type of sleep that is not described.
Had not considered that.
Neither non-REM or REM.
So what I'm getting is that the sleep disturbance is now a consequence of the pathology, because earlier we're talking about how poor sleep would exacerbate, but now when you're this far into it, it's making your sleep worse, right?
Mm-hmm. And I do think the sleep deprivation that ensues, because I'll say more on that in a second, the sleep deprivation that ensues then leads to, it's a compounding effect.
Right. So is that known in people that, Jason, do Alzheimer's patients sleep more and more poorly? Is that known?
Yeah, I think that is a symptom.
Okay.
A lot of these things, it's just never clear what the chicken and egg is, where it's sort of, you get pathology and then you have sleep disruption, or is it you have some sort of sleep disruption first and then you get pathology?
Or both.
So.
Yeah. And that's kind of like at the beginning of the episode I said, I didn't say chicken and egg, but the way they present the story is that it's the hippocampal dysfunction that occurs first, and that this brings on the something's happening in the hypothalamus, but it could it not also be, in a different direction? like I said, there's, I know that like most of your nervous system is actually in your gut. I mean, but there's a lot there. So me saying that like, there's gut problems that are happening in this prodromal phase is like, oh, and didn't we talk about some paper that's like stuff going up the vagal nerve from the gut to the brain? Or am I just
in Parkinson's disease? I think there's a clear connection.
But so I wanted to say something about like what Tim said about potentially a new kind of sleep, or just like, we talked about the different types of oscillations. There's this like adaptive effect, kind of like a recovery effect in rebound periods of sleep, where you enter slow wave sleep much faster than you typically do, and you stay there for longer. And so it turns out that the APP mice, they don't have that. So they do not spend more time in that like delta slow wave sleep period. So they say that there's a selective reduction in REM sleep. Yes, that is true in sort of, because the non-REM, which they've kind of crammed all of these different states together, the non-REM is actually affected, but it's just, it's not, and it is reduced. There's a part of it that is reduced. And so while, REM might be involved, it's this oscillation between REM and non-REM that seems to be important for that hippocampus cortex communication, that we're starting to understand more about. So if REM is disrupted and then there's pieces of non-REM that are also affected, then like what role does delta, what role does slow wave sleep play in consolidation? And that's just something I just don't know, but it may be out there in the literature.
Yeah. No, I think, well, all these different sleep phases have been implicated. I think it's not super clear, but there's also this rebound effect is also considered like a homeostatic effect that, sleep deprivation induces the sleep rebound, and that seems to be disrupted. So, but, I feel like this, as you said, this is kind of an isolated observation. They don't ever really directly then connect this to the hippocampal plasticity or synaptic function.
Right. I mean, it's tough. I was thinking about this a couple of weeks ago because there's somebody here, I have a colleague who studies the induction of there's like on and off states in slow wave sleep, and they're able to induce off states in waking mice, and they're trying to see how that's impacting cognition. And so I was thinking like, man, like it's kind of tough to like how would you change a mouse's sleep architecture experimentally? Like how do you make their brain go into one, to have more, gamma oscillations.
When we won't get into ripples and things like that.
Oh yeah, no. Some other time.
I guess the final experiment that they then also sort of, I wonder if this is even a reviewer question.
Oh, did you look at the, did you think it was a reviewer question, or did you actually go into okay. Cause I think they did make like some of the review process stuff available, but I didn't get a chance to look, but yeah. Okay. Go ahead.
But now they were like, well, is this relevant to humans? And so they finally.
Oh yeah. Show us in Alzheimer's disease brains, like show us something. And that something that they show is, again, getting back to like, well, what's really happening to the MCH neurons? And does it actually like precede, some of the things that are being portrayed here as being the drivers of the cognitive decline and the changes in excitability. But they look at the trafficking of MCH. So they look at the trafficking of MCH in the axon. I'm assuming that they looked in the hippocampus. And so I'm not sure how, like, if trafficking is really the right word, because we saw that the transcripts were inside the axon. So anyway, but in any case, they say, they're looking at the accumulation of, or the puncta that are that's MCH being transported along the axon toward the pyramidal neurons. And they find that there are no changes in the number of MCH puncta in the axons at 3, 4, 6, and 9 months old. So at 6 and 9 months, the animals are like overtly affected. And then at 4 months, we know that there's an increase. You would maybe expect there to be an increase in the number of puncta at that 4-month time point, given that they saw that there was an increase in the transcripts. But sometimes we don't have lining, the protein and the mRNA don't always line up because there are other mechanisms that control translation and mRNA stability, et cetera. But they find that at 6 and 9 months, the puncta size, the MCH puncta size increases. And so they're thinking maybe there's some sort of accumulation and,
I think of it as constipation of
axon.
MCH peptide, since you're talking about prodromal constipation.
That's a good one.
It's all blocked up, so you have these giant puncta and they can't release it. And therefore you don't see the compensation to shut down these neurons because all these MCH is like stuck.
So then if that's true, like why is the trafficking affected? Because that's not something I would typically think. Like if there was something happening, like the changes in the activity in the hippocampus, if that's projecting back to the hypothalamus and changing hypothalamic neuron function, which then goes back to the hippocampus, I just wouldn't typically think of like changes in trafficking necessarily being like the first thing that would change. But
do you like this? Do you think these data are convincing, or could it just be that this is stuck because the brain is getting messed up, structurally?
I mean, I think it's super interesting, and they just didn't have. They didn't have time.
They have some correlational data where they show that if you label for these amyloid beta plaques, then these amyloid beta plaques start forming, maybe before 6 months, but at 6 months when all these things start falling apart, these constipated MCH
in humans
nodules, these like blow up blobs, they are stuck inside these plaques. So maybe these plaques impede them from being released.
But it's definitely correlational, and
and it's definitely true that these plaques act as a physical barrier and really end up rewiring the axons and projections in that area. So I think the key that's just very difficult to really determine experimentally is what's the effect on the release of the peptide. And they just that would take another kind of tool.
And I think that this work is ongoing
increasingly.
Yeah, of course.
Go ahead. I was going to say lots of fiber.
So since these peptides bind to, I think, receptors that are G protein coupled, we can look at, we now have optical sensors that are G protein coupled receptors whose fluorescence would change depending on whether things are bound to it.
I just don't think anyone's made that for this particular.
No, right. So it needs to be made.
It's doable.
It needs commitment.
I mean, I think that's, given my own personal experience, and I'm sure the rest of you, there are times when you just don't have like the paper needs to be published, and that there are things that are missing, but you can't. You just
do everything.
You can't do everything, and like there's your career to think about. Like you got to get some papers. So even though it's not like the full blown like Cell paper that that it could be, you just don't have the time. Sometimes you don't have the finances.
This is already a ton of work. So I think it's easier for us to in our chairs here to say, well, why did they do this experiment?
Oh no, I fully recognize that.
They have human brains. I mean, that's already above and beyond.
No, I fully recognize that. It's great.
Vivianne, can you give us the summary now?
So the summary is put on your Apple Watch or your Samsung Watch and track your sleep. And if you see that your REM is disrupted and there's no other reason, then go out and, I'm sure that the vitamin shop or whatever, they're probably selling MCH at this point because like we should
Well,
just supplementize everything.
Well, they say it's not a good idea to take MCH.
You know, I actually appreciate that that they said that, because there's some other situations where it's like, oh, maybe this thing could help. You should just take it and see what happens. And I'm like, oh no,
yeah, right.
Like
right.
You have no idea what that's doing, and it's like just a thing that you could buy at the supermarket. So no, but I think the takeaway is that there's like, we may now have at least part of the mechanistic picture that links sleep disturbances, that links sleep and memory and cognitive decline. And we've brought up many questions here that, just were outside of the scope of the paper and were probably the next papers that we're going to see coming out of this individual's lab, if and when she, starts her own lab. So, or her colleagues. So I think it's great. And their abstract was really well written. So that's just the cherry on top for me. It's
they also have a nice model figure in the extended data that explains all this. But as they say, this impaired MCH dependent function, it synergizes with perturbed REM sleep, and that makes it worse. And then you get the aberrant neuronal activity, and then the seizure susceptibility. So that's
they did have a little thing about the threshold to seizure induction was lower in these animals.
And that's been shown in other mouse models of Alzheimer's. I think this hyperexcitability phase is really key, and has been shown also in humans that there's these subthreshold seizures. They don't have full-blown seizures, but there's something going on there where it's clearly not normal.
That there are ictal waveforms, but no overt seizure. And I think that's been seen in bipolar disorder. So, and then the last two words of the article are psychiatric disease. Right. So it'll be interesting to extend this to those studies.
Is this the first time MCH has been shown to be involved in Alzheimer's?
I think there was a paper that I put a bunch of stars around that was treatment of mice, let's see, what was it? That you can recover. Let's see what it is. Rescue of memory impairment with MCH administration. Yeah, it's reference number 74. Nasal cavity administration of MCH improves memory impairment in Alzheimer's disease mouse model. That was in 2019 though, so that's pretty recent. So this might be the second paper that shows it in this way, but there's some other papers in there that are it influences the hippocampus, but not necessarily in the context of AD.
Yeah, and the sleep connection is definitely really interesting too. So,
All right, cool. That's great. Thank you, Vivianne.
Yeah, my pleasure.
Very nice. Very, very thought-provoking. All right, that is TWiN 41. Show notes, microbe.tv/TWiN. Questions, comments, twin@microbe.tv. If you like our work, consider supporting us, microbe.tv/contribute. Jason Shepard is at the University of Utah. Jason Synaptic on Twitter. Thanks, Jason.
Thanks, and thanks, Vivianne. Yeah, that was a fun paper. Look, right down my alley of lots of things that we were sort of interested in as well.
Yeah. ARC definitely popped up a few times in there. I saw that.
Tim Chung is at New York University. Thanks, Tim.
Thanks, Vincent. Thanks, everyone. Thanks, Vivianne.
And Vivianne Morrison is at Tulane University. Thank you, Vivianne.
Yeah, no problem. It was fun.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.